to part 3 of the Axovant analysis. This video will separate, there's an old phrase which is a little bit sexist. It's called separate the men from the boys. So we could say separate the women from the girls. Either way, the point is that, a lot of people can analyze clinical trials and they get an inkling of the right direction, but not me, not Martin Shkreli. I make sure that I'm right. And you might say, how do you make sure that you're right? That sounds impossible. Well, it's not impossible. I'll show you. First thing you should read is my paper on vital therapies, which I wrote. It's got a few thousand views here. 44 pages. Pasted in chat. It's called The Sisyphean Odyssey of the ELAD, Why We Think VTL is a Short. It's written by two of my colleagues, Chris and Lewis, and of course myself. I did most of the writing of this. August 2015. And pardon me while I go in here real quick. But basically, I'll show you what happened. I made about $20 million on this day in August, late August 2015, where I, in essence, predicted that a clinical trial would fail, very similar to the one upcoming from Axovant. And I'm not saying that I'm predicting that quite yet, so we're not quite there. Let's do that. You can see the trial failed. Stock went from 17 to 3. 17 to 3. That was a very happy day. Ironically, it was options expiry Friday. So options, the company came out with the news after the close on Friday, so I had to exercise all my options. And it was quite the day. It was nice to make $20 million in one day. The stock ended up recovering a little bit, but I think it's back down to 3. Just goes to show you, you got to think about the move after. It's possible that the stock just was gonna. I'm not good at these charts things, but let's see here. It's possible that instead of the stock going from here to here, the stock could have just gone from here to here, and if you had, which is what, at one point it got up to $10, so it only went from 17 to 10. You have to plan your short out very differently, very, very differently than if you suspect it'll go from 17 to 3. Now, I was lucky enough, it actually took me several days to cover, as I was short many millions of shares through Delta exposure and things like that. I was short well over 1 million shares, and you can see the liquidity wasn't so fantastic, so I didn't want the stock to go up too much. But I got out, and that was that. So, in any event, you have to search for either Interberdine or you have to search for it's Axon 101 or RVT 101. Anyway, so that's sort of so anyway, if you read this paper, the reason I mention this paper is not just to brag. It's to sort of, especially the first 2 or 3 pages, talk about philosophy in many ways. You have this philosophical concept of uncertainty, which is both statistical and philosophical. I even quote Bertrand Russell here. It says, when one admits that nothing is certain, one must, I think, also add that some things are more nearly certain than others. And refer to Kant and Hume here. Again, I'm not doing this to brag about my intellectual, credentials. I don't have any. I'm doing this to simply sort of explain that, to try to try to prove something in the stock market is possible. You just have to sort of have an open and mathematical mind to it. So anyway, if you look at this, basically there was this conditional analysis, which I think made a lot of sense. So basically, I like to call it the multiple condition kind of analysis. And basically, it just means that conjunctive low probabilities will often result in an impossible outcome or a certain outcome. So I'll sort of show you an example here, and we can start to do it for Axovant. So anyway, what are the conjunctive low probabilities? Well, first we need some events with low probabilities. So event A, event B. All right, so event A is, let's look at our model real quick. Event A is that mindset will recapitulate the one or the most successful of the 4 clinical trials that GSK ran for intepirdine. And so that obviously, I think, intepirdine. That obviously, I think you can imagine, I mean, it's almost random, right? I think it's less than 25%. At best, it's 25%. So you're basically saying the outlier, right, is going to be the one. This is the study, so-called study 2, which is also the biggest study, mind you. Saying that this study will be the one that's recapitulated, but you know, that's sort of like saying if I shot 9 out of 10 free throws on Monday, 6 out of 10 on Tuesday, 3 out of 10 on Wednesday, and then 5 out of 10 on Thursday, that the next time I'm going to shoot free throws will be 9 out of 10. I think that's obviously, I think anyone with a brain will tell you that, look, your average is 5.75, not 9. And it's unlikely that you're going to shoot 9 free throws. Is it possible that you shoot 9 free throws? Sure. But if you said, if we did the same experiment, I think I get these numbers right, and I said, okay, you're going to shoot 1,000 free throws, you would really bet against me shooting 900. So I think that, you know, that's just sort of something to consider. So the probability that it recapitulates the very best study, I'd say, is far lower than 25%. So event B is that I would say that intepirdine has positive efficacy, while allopiridine, idalopridine. By the end of this, we're going to learn how to say it right. Idalopridine. You say the erdine independent, right? Intepirdine and idalopridine. Idalopridine. It could be ida or ida. I would say it's probably ida, but it could be idalopridine. And this is definitely intepirdine. Cavefamidine. That's a good one. All right. So anyway, intepirdine has positive efficacy, while idalopridine does not. I'd say that the probability of that happening is very low. You know, usually if you have one drug, there's a class effect amongst the other drugs. In fact, class effect was preserved in acetylcholinesterase inhibitors. For example, donepezil what was the other acetylcholinesterase inhibitor? Reminol. I forget Reminol's generic name. It is galantamine. May not be acetylcholinesterase inhibitor, actually. It could be. I don't know. Yeah, it's a cholinesterase inhibitor. Galantamine. Acetylcholinesterase inhibitors. So you got Aricept, you've got galantamine. I think there's one more. One more FDA approved one. Anyone know? Might be this one. I don't think so though. No. Definitely not. Rivastigmine. Exelon. There you go. So all these drugs are FDA approved for Alzheimer's. And none of them didn't work. So it'd be really weird if interberdine worked and, let me see if I could turn this up. It'd be really weird if interberdine worked, but idalaperdine did not work. So we know that idalaperdine failed. Failed 3 phase 3 trials. It is rare in medicine. It is very rare in medicine that one medicine will work and the other one won't in the same class. And we know that, in fact, if you look at the physiochemical properties, while this drug may partition quite a bit more to the brain, it still is a similar nanomolar, really talking about a 3x potency, so almost exactly the same potency. In fact, I'm not sure actually. Let me take a look. Well, we know it's subnanomolar. We'll look up the idalaperdine Ki in a second, but it's very weird that those two. So anyway, if you multiply the opportunity, so you basically have to get comfortable with both of these events, right? So both of these events have to take place. So you're talking about a 2.5% probability of success. So that's not quite 100%, but remember this 2.5, this 25% is generous. And the 10%, may or may not be generous, but you'd have to overcome both. You'd have to have an independent. These conjunctive probabilities have to be independent. This has to be independent from that, and I think they are. Basically, even without the phase 2 studies for interberdine, if you ask me today, design a new 5-HT6 antagonist, right? Would you do it, Martin? I would say no. I know from the idalaperdine studies that these drugs don't work for Alzheimer's. I would not do that. Lundbeck ran 3 big phase 3s, right? They ran Starshine, Starbeam, and Starbright, and they all failed. So why would I want to do that all over again? For a while, it looked like idalaperdine had bad safety. But it turns out that its safety is just fine. It's efficacy that doesn't work. So the idea that interberdine, totally independent of what we've seen in the clinic so far, would be at odds with logic, right? And so if you look at the studies themselves and forget about the mechanism, I would say that it'd be at odds with reality as well, but independent of its mechanism. that of these 4 studies that we'd see the very best result get recapitulated, which is only a 1.6 point improvement. We got 0.4 improvement here, we got 0.128 improvement here, we got a 0.7 anti-improvement in essence here. So the average of that is very bad. So it would have to be 1.6 for the trial to be successful. And we did a little bit of statistical analysis here where it's at the bare minimum they would need a 1.5 point change to be statistically significant. So and that's basically the best result they've ever seen. So we'll I think that we're at a pretty sort of bad state of affairs for Axovant here. Now the only question is, can we get more feedback and feeling as to why this is a good idea. For example, event C is 5-HT6 antagonists are a bad mechanism of action. Now, is this an independent event? I don't know. It sort of seems like a sub-event of event C, of event B, I should say. So we can come up with another sort of candidate event. And a bad candidate event, which we'd reject, would be something like, well, GSK passed on it for a reason. That's a stupid sort of candidate event. GSK doesn't know more about Alzheimer's than we do. It's not like they're any more of an expert than Axovant is, or that we are. We don't really look at that as a legitimate way to think about it. So, but it would be an independent reason, as maybe they know something, right? Maybe they know something that we don't know. Any other candidate events? So obviously, again, just to be repetitive here, for an event, we'll call it, I don't know, let's see. Probability of some event X is the conditional probability of sub-event A multiplied by the conditional probability, as long as they're independent, of sub-event B. In other words, the probability of I don't know, lightning striking in New York and there being an eclipse in Japan, or the two have to both occur, so the probability is one multiplied by the other. Maybe somewhat similar as if I flipped a coin and I rolled a dice. The probability of heads is 0.5 times, and the probability of, say, rolling 1 on the die is 0.5 times 0.16, which is a lot lower than it might seem. It's only 8%. So one of the things that is a little bit tricky about statistics versus when you kind of look at something through a practical or you think you're being practical lens, like, oh, this drug might work. But if you actually sort of take a step back and say, let's actually really try to get our heads around this, it's actually much harder to get a new drug than it looks like. So in any event, if you can come up with another sort of candidate event, I'd be interesting. There's rarely more than 2 or 3, but I'd be curious. But we should break these down really carefully, because if they can't overcome these, we basically we can say we can prove the null hypothesis. The null hypothesis being the 4 trials are not to be counted on. In fact, one of the trials had such unpredictable events, or such reliable results due to the donepezil failure to separate from placebo. This is trial study 1. We'll just say NCT 0708552, that it cannot be counted. So at worst, the 3-trial mean change is, you got to be your own devil's advocate. You can't be curve fitting for your own benefit, but one is 1.6, 1.3, and 0.4. So if we take negative 1.6, negative 1.28, and negative 0.4, the average there, each one changes negative 1.1. So it always bothers me when you throw out the outlier. It's sort of like saying, you know, it's kind of hard to throw out the worst result and say your new mean excludes the worst result. If you do that, you should also throw out the best result. But if we take that for granted, I'm still not sure that that's going to work, because you can see that to get a 1.1 statistically significant change, you would actually need, and we could do the calculator one more time, for a 1.1 point ASCOG, the standard error of 0.43, the p-value is 0.07, so it's still a failure, even if you throw out the worst, the very worst trial of the 4 trials. You'd have to get down to a standard error. Let's try it with 0.4 standard error. And this is assuming no dropouts, which we know is unrealistic. So this is still a failure at 0.052. 0.4 standard error. So pretty tough position to be in, I'd say. So even if, you know, the null hypothesis is true, you're really at a very risky situation. Now, this thing, I think we've got kind of dead to rights. I mean, idalopridine did not have efficacy, it did not separate from placebo. So you tell me how interbidine is going to work when its cousin didn't. So we're going to have to sort of look at the literature. I think what we can do here, again, is sort of what separates the adults from the children, maybe the best way to put it, is to sort of do these, to get to be very specific, granular, and careful. Let me try to do that. So we'll come up with some search terms. I think the first search term we used was, there's 3 right here. So let's use these. And then we want to use the Lundbeck ones, right? All right, so let's start with RVT 101. See if we get any results. We got 12 results. Let's see if there's this one, Bella. All right, so that one isn't there. How about Calhoun? No? All right, well, why don't we just take these and take the output. What I like to do is to paste them. And this is a little cumbersome. Again, the job is not all glitz and glamour. Sometimes it's really painful and slow. fact, it's always painful and slow. But you gotta grind through it. I didn't say it'd be easy. Making lots of money really fast is very hard. It's worth it in the end. We're looking for the bet to fail. That's exactly, this is a great bet. You make a fortune when the trial doesn't work. I'll go through the ethical ramifications of that maybe later, but if you're about your paper, you can, this is as good as a success. So I'm looking for doubling of papers. So you can see the Higgs paper here is doubled. So I'm gonna remove that. Let's see if any of these other ones. The de Bruijn paper is not doubled. It doesn't appear to be. Just want to make sure that I'm not going to read the same paper twice or whatever. We want to kind of know how many papers we're up against here. How about this Jejerian paper, effect on olanzapine on scopolamine-induced deficits in differential reinforcement of low-rate schedule in rats. That is a double. The second de Bruijn paper, effects of risperidone, clozapine, and the 5-HT6 antagonist, GSK, PCP-induced learning defects. Give the mouse PCP. It's crazy. Oh, this one, Callaghan. It's only one Callaghan paper. All right. Like I said, this is going to be painful. Fidget spinners on sale for $14,000. All right, here's the Maher Edwards paper, SB 7457, and donepezil. That's doubled. Oops. The second Maher Edwards paper, double-blind, placebo-controlled study. Look at the PubMed ID. That's how you can tell it's the same. And here's the Idris study, which is sertinidol improves. It's not the same. There's the same PubMed ID. Okay. So there's sort of, 6 unique papers here. Calhoun, Ferraro. I think that de Bruijn paper, the selective. Okay. Parker and Callaghan. Okay, cool. So we got that search term out of the way. Let's do the next one. And sometimes these hyphens kind of mess things up, so let's see if the RVT without the hyphen will do it. Nada. Alright, so let's try GSK 742457. See if we get any results. We get 11. Alright, so let's do the same exercise. Let's see, Calhoun. This is the same PubMed. Higgs. This is the same. How about Parker? Over here. Jejerian. Got that one. Brown. Yep. Callaghan. Same one. de Bruijn. No? How about this one? No? That one. Okay. Maher Edwards. Yep, that's that one. Second Maher Edwards. Idris. Yep. Now Mork. I don't think we saw Mork. No, there's Mork. Alright, so all of these are recapitulated by the 16 one. So let's see what we can do. I think we can just remove this. Let's also just add the fact that we did that one, and then let's try to do no space here. No documents, good. And then let's try to do just the number. Sometimes that helps. Okay, that doesn't get us anything either. Although we're getting the UUID, so let's not get the UUID. Let's see if we can do that. Okay, that actually gives us an interesting result. Let's try it. Like I said, painful, slow, but necessary. Alright, we got the Zajdal. Oh, is this the same exact list? Zajdal, Grokowski. So this is the same result. Ivanchko, Higgs, great. This is the top result in exact order. Kodani, great. Wait. There's no Rossi. Maher Edwards. No, there is Rossi. Okay. 13, Maher Edwards, Idris, Mork, Upton, Twang. Alright, so this is actually up here. 4, 2, 4, 5, 7. Alright. Let's try SB. SB stands for Smith Klein Beecham for anyone who doesn't know. Alright, let's try that. 17 results. Alright, we got the Calhoun. Zajdal, Grokowski. Oh, this looks like the same stuff again. Parker. Jerian, DeBrown. Callaghan. Witten. Scared me. Let's see a Whitman. Oh, there's a Whitman. Cadoni. Liam Moliner. Maher Edwards again. Rossi, Maher Edwards. Idris and Upton. Alright. So all of these are subsumed within there. So let's try it with no hyphen. We get 10. Actually, maybe some novel ones here. Let's try it. Alright, Calhoun, we got. Higgs, we got. Parker, we got. J. Arajan, got that one. DeBrown, Von Drimlin, Callaghan and Hawk, Liam Moliner and Rod, Maher Edwards, and Idris. Alright, so all those are subsumed as well. Cool. Just about done. Now we're going to look for the Lundback. So let's take that Lundback search term. 16 results. some of these might be different because it's a different drug for the first time that we're looking at. Let's take this guy, break it down here. All right, De Jong. That's the first. Looks like the first De Jong paper. Let's try looking for the PubMed ID. Yep. How about this guy, Ferris? Yeah. These are all novel new papers. Great. Editing rights to my models? That might be very tricky. You can comment. If you email me, maybe I'll give you editing rights. You gotta prove you're worthy. Alright, let's see for this one. 282538. Alright, so we got the same one here. How about Mork? Alright, it's a novel Mork study. And Mork is a Danish researcher, so he probably works at Lundbeck, is my guess. Lundbeck, of course, a Danish company. Neuroscientist experts. I have some friends who worked and work there. It's important to get a network going. Alright, let's see if Mullard looks like a brand new paper. Kuczynski looks new as well. Let's try this. Kumar. This is probably not relevant. Alright, Vanchico. Oh, there's duplication there. Dudek, or Dudechk, in the language. Obesity, that'd be interesting. Alright, another one. Galimberti. Almost done here. This is the latter study. I'll have to look at this very closely. Witten. Witten's double. Okay, Arndt. Alright. Got a whole bunch of papers to read. It's not a terribly large amount. Let's do one more search for idaloperdine. Before we do that, let's make sure if we add a hyphen, get anything different. Same 16 it looks like. Yep. Sometimes you have to be super careful. Leave no stone unturned. Now let's have no hyphen at all. I think this should be same results. Just eyeballing them, it looks like they're all the same. Mullard, Arnold, Mork, Calhoun, great. All right, let's do the final search for Idol Alferdine. You'll see that this is not exactly the final search. We'll go through that in a second. Oh, look at this idiot. Surely you could write a program to do this for you. Yes, the program will predict every 5HT6 antagonist. Interesting. All right, we've got this one, De Jong, Ferris, Calhoun, Mork, Ferrero. Impatience of some people are so incredible. You know, we've been working for half an hour and you're already tired. Can't make money that way. That won't work. Not one bit. Mullard. Wachikowski. That's a new paper. Kuczynski we saw, I think, yeah. Harik. Kumar. Godin, that's a new name. Alright. Dudek we got. Galimberti we got. Wilkinson we got. Schneider we got. Arndt we got. Alright. So it looks like there are 16, 6, 22, 13, 35, 36, 37 papers to read here. I want to do one more kind of wild search. It's a little bit too much, but I'll show you how to manage something like this. 5HT6. Alright, we got 533 results. So what we'll do is we get per page, we'll get 200. That's actually less than I thought there'd be. Then what we can do is Alzheimer's trial. It's a lot. Maybe we do phase. Try donepezil. That's a lot too. Who would have thought people researched Alzheimer's disease? Donepezil trial. Wow. Holy smokes. Alright, well we can look at those papers at some other point. If we wanted to. So one way to do this is you just do some So we have our literature set up, and we should start printing some of these out. So we should start maybe with the Zajtal paper. I reconnected my printer today. Alright, so this is a MedChem paper. It's not going to be too useful. Like I said, this is, we looked at this briefly the other day. This is a bunch of, it's a medical chemistry department. Alright. Pretty sure I should be able to read this. If not, we go to our old favorite, which I would never encourage you to ever use. I'm using this on my own risk. We can save it as well. Hopefully my printer will wake up. Then we're going to go to USPTO as well. Here are the... I didn't think AVN 211 made it to trials. It's AVN 211 Avenuro. Yeah, we should probably study that. Oh, it's a sulfonamide as well. Isn't there a Pfizer 5-HT 5-HT6? Another MedChem paper. We'll read them in the reviews, but I think there's a Pfizer one. So one of the things we're going to do is we're going to go to USPTO.gov. We're going to search for patents. So here's the main patent site. We'll do this sort of a similar maybe search for 5-HT6 antagonist. And this is going to get a lot of results, unfortunately, because I think what it does is it, oh, maybe not. It's a wrong hyphen, that's why, 5-HT6, that's it. It's like programming, you gotta get the exact letters down. And this is far less, excuse me, this is far less than I thought, actually. So what we can do is we can copy pasta this. See if this will take well. Well, perfect, in fact. We put the assignee in here. Is that how you spell assignee? I think so. Yeah, you assign something with two S's, right? Now, I'm wondering why my printer's not working. That's getting me angry, getting me all heated, getting hot under the collar here. I reinstalled all my drivers. This printer's making me real pissed. Alright. Let's put the assignee in here. Alright, so the assignee here is Lundbeck. This is Ono. Different target though. So if we see Lundbeck, we don't need to do much, right? But if we see a different company, we should stop and look. And you can see here that this is not a 5-HT6, this is an ENPP2, which actually could be useful for me. Some rare diseases with this illness. I don't know why Lundbeck would have one. You learn a lot by doing this. You can go to Lundbeck and buy this drug for probably nothing. All right, let's see number 4. Again, Lundbeck. Medivation. Huh. Interesting. This could be Dimebon related. Dimebon was an old failure. Let's see where they mentioned 5-HT6. They mention it pretty vaguely, so non-specific. So this doesn't look like an interesting patent. Let's keep going. This is Lundbeck. Ono again. Is Ono interested in these? No, it's not. Alright. Get a lot of these continuation and part patents. That's what they're called. So these are all CIP patents. See this Dr. Hung as an inventor here. Like I said, they had an Alzheimer's drug. It did not succeed. 5-HT6. This is a method patent for Down syndrome. This is a method of use patent, so it's not too particularly useful. But it's the first somewhat even remotely interesting patent. I'll mark it down. It's the Ono patents again. ENP2. It's the Medivation again. Or no, this is the Ono. Tetracyclic, is this ENP2? Yeah. Okay. Bridge heterocyclic compounds, Medivation. Now let's see what this one is. Medivation. It's Medivation. Galenia. Ooh. Galenia. That's so weird. It's a composition of matter patent. Let's see if it's a 5-HT6. Huh. That is so funny. So, Medivation looks like, so the David Hung's former company looks like they were focused a little bit on 5-HT6. Let's look at these patents actually. I wonder if that's how they think Dimebon worked. We should also put down Dimebon, actually. Make a Dimebon page. Dimebon, for any of you who don't know, is what Medivation was studying for Alzheimer's disease, and that trial failed. Medivation ended up being successful because of their cancer drug, though. I don't know what this is. Suvin Life Sciences. Oh wait, I thought I was looking for the HDAC. What am I doing? This is 19. Isn't that weird? 19 is that, and 20 is HDAC. Okay, good. LixT Biotechnology. HDAC has nothing to do. That, this is Medivation. Okay. This is Galenia. Let's try this one. This is the Suvin. And notice the sulfonamide creeps up again. I'm not sure what these Suvin guys do. It seems familiar. It's definitely interesting that it looks like they've made novel compound. Alright, let's try these. Medivation again. Tetracycles. This LixT thing again. HIV, what? Oh, these are all still HDAC. I'm so confused. It's like jumping around these links. See, it's the wrong. It's going to the wrong thing. Isn't that weird? Let's do a search again. That's so strange. I'm trying to open this patent, 8138, and it's giving me 8143. Oh, broken website. Garbage. Alright, so this is a novel, 5HT6. It is the government, isn't it? How about this one? 83, yeah. Again, I'm trying to open up 8304 or 431. And it's not pulling up the right damn thing. This is Pharma Neuro Boost. Weird method of use thing. How about this one? Now I'm worried I got them wrong. Some of the links look corrupted at least. Okay, this is I think the continuation in part one, basically. Method of treating binge eating disorder. Got that. Protective agents. Nope, that one didn't work. This is the Hung patent. Let's see if this opens up correctly. Nope. What a waste. Might as well search these one by one. Probably be faster. Alright, this is Wyeth, so this is big. This is Pfizer basically. Go back up here. I knew Pfizer had something. So not everything's published, but everything is patented. That's the lesson in this. All right. Brain Cells Inc. Sounds promising. Not. Yeah, basically when you get one patent, you get a bunch more because they come in with different claims. We're about halfway done here. It won't take too much longer. D2E. This looked like I was predicting would be the Lundbeck patent because of the fluorination. Let's see. Target. Oh, it's a time of bond derivative, maybe? Process of losartan, that's funny. Talk about our element. Angiotensin. A lot of funky stuff here. Yeah, it was a good instant quote. Alright, benzofuran derivatives. You guys know what a furan group is? Pretty simple. So benzofuran, you could picture what that looks like probably. Biovitrum. Look at that. Benzofurans as 5-HT6 antagonists. No benzofuran groups here. Well, I've always been an investor, whether it's medicine or stocks, they're sort of one and the same. Oh, look at this, Roche. Big boys. Oh, Bonhouse. I know this guy. I know this guy. Aha. Aha. Some of the best medicinal chemists out there for these kinds of drugs. Memory. This was Roche as well. Memory ended up becoming Roche. These are all bullshit patents. Here's a real one. Oh, there's the Wyeth again, aka Pfizer. More Wyeth. Korea Research of Chemical Technology. All right. Quinoline diones. Kind of interesting. Look at those structures. Wyeth. Okay, stop distracting me. You get banned if you're not talking about the topic at hand. It's starting to tick me off. So it looks like Goliath was there. I think GSK was probably there first, but let's keep looking. Roche. I don't recognize these chemists. We have another Roche group up there, but they're different chemists. Ship of the type. I love these old patents. This is from probably the 60s. 1975. A ship comprising a hull. Those inventions, though. Alright, get some more live patents. Almost done. Eli Lilly. Throw their hat in the ring as well. We got Roche, Lilly, Pfizer, GSK, of course, Medivation, and Lundbeck. It's almost always going to be crowded medicinal chemistry space around any serotonin target. They've been such successful medicines. Yeah, that's right. BioVitrum as well. Don't forget BioVitrum, which is now Swedish orphan BioVitrum. Wyeth. Shout out Kitty. These have to be more than one chemical series from Wyeth, as they span all the way back to 2006. So I think Pfizer might have been here first, although GSK has to show up too. Shut up. Yeah, I've never had brunch in my life. I don't do brunch. I'm a grown man. Brunch is for women. Syntax. Syntax was Roche, for anyone who doesn't know. These are old Pfizer Wyeth compounds when Wyeth was still independent. The only question is, where the heck is GSK? Let's do another search. There's SmithKline Beecham. I'll just write GSK. For ADHD, that's funny. Hitting this website too hard. There we go. 2002. Beecham. It's Beecham. As I thought it was. Alright, so we got some space here. Let's do another search though, because I want to be, like I said, really, really accurate. Alright, so it's 587, these bad boys. A little bit more than we bargained for. But that's life. We're not going to search them all right now, but let's just paste them. Some people aren't as funny as I am. I'd say most. Practice your comedy somewhere else. I'd be curious to see what the first 5H26 patents were. And of course my paper still hasn't printed. Something's wrong with my printer. Spent half the day today messing around with it. I guess use the one in the office, but the one in the office kind of stinks. A better one too. I need a copier. We've got two offices with a copier in one of them, not in the other. I don't know you, Tom Phelps. Go away. Someone ban this guy. I'm too busy. Thank you, my trusty trashy assistant. No trashy. My brain is. Alright, so we don't need the knockout mouse one. Aha. Merck. Merck had the first. make sure we're getting the right one. I'm nervous about these numbers. Aha, Smith Klein Beecham. Let's see, that's 6380. Is that 6380 the one we had already? I think so. Yeah, 6380, 199. 6380, 199. Alright, how about 6403? Stupid thing. Virginia Commonwealth University, there you go. Sulfonamides. Alright, 6423. Okay. Sulfonathiloxyl. The tongue twister. Sulfonaloxazolamines. Shut up, kitty. You stinky. Oof, you stink. That breath stinks. Ah, Pharmacia. That's an oldie. Look at that. Talked in 2002. NPS Allelix. What the heck is NPS Allelix? Never heard of it. These are interesting, some of these old patents. You can kind of see pharma sort of gave up on this line of inquiry. We have this patent already, 6627661. Yep. What a memory. Benzofurans from Merck now. Merck Wyeth Pharmacia race for 5-HT6s. A race that everyone would lose. That's how chemistry departments work. 5-HT5. Spectrum Pharmaceuticals. What the devil? Let's see every big pharma in here. I'm getting back to the broken patent search. 246. I wonder if these are it's linking because it's a continuation of part. Or is it linking because it's broken? Merck DE, which is different from Merck. All right, well let's. This is interesting enough. But the big question is, did any of these companies put these drugs into the clinic? I guess we didn't search for 5-HT6 antagonists in here, so let's do that. Let's try that. Let's see if my printer will work someday. Wish I was a network expert. Try restarting this one more time. 27. Alright, so let me put these here. We'll do 6%. Kunasawa. There's 2 Kunasawa papers, same paper. Do that. Oh right, it's printing. This is the Avid Neuroscientists. Let's see here, 260. Well, everything in here is going to be in there, so let me actually go about it a different way. Let me delete these. Here's another 5-HT6 antagonist, PRX 07034. From Epix. Epix was acquired by somebody, I forget. I remember Epix, the public company. 253. Abraham. I guess GSK had two of these. Phase 1 study of interbreeding. I wonder if the Wyeth compounds did make it to the clinic. There's a publication on it. I thought Pfizer had one, but it could be crazy. There's 27 results. Forgot to paste the rest. I'll save this model in my public folder, so y'all can view it and leave comments and stuff. You're welcome. All right. How shall we do this? A handful of articles here. I'll just slide them in. Alright, so now we have an organized sort of corpus of literature to look at and study. It's a lot of work, but like I said, that's how it is. We start with this, printed on 9/12/17. Let's see if we can get Gruchikowska or something like that. Or did we get Gruchikowska? I think we got Zajdel. They're the same people. Are they? No. No, they are. There's Zajdel and Gruchikowska. That's so funny. Alright. Yeah, welcome to the real world. There's lots of homework. Come on, Sci-Hub, do your thing. In the meantime, let's get this guy. This guy's doing, he's got like a million of these. Chukotchenko. I guess ACS, I just generally don't have a availability. I wonder if I have an ACS login, I might. Now let's go to number 4. Alright, at least this one's free. Hopefully that'll print, although it may not given my printer. But at least I've got the PDF. Try this one. Okay. Looks like they did a 5HT6. Oh, it looks like a 5HT2A antagonist as well. What? That's important. That's very important. say at least that I saved them, so I don't need to save them again. Let's get a couple more, and then I'll save this file and we'll call it a night. Huh. Interesting. This is the tracer paper. We might have read this one. Alright, so let me see if I can save this stuff. Just bear with me while I do that. I gotta do some sensitive file switching and sharing. Very sensitive. Okay, we're back. Alright, let's just save this. We'll first share it. Get shareable link. Anyone on the web can comment. Great. Done. Alright, now I move to models, move here. Okay, so you guys should be able to use this and make your, I think you, I wonder if you'll be able to copy it. I think you might be able to download it. If not, let me know. Why is this here? Why can't I view? Is that strange to you? After O, it's all messed up. Why do you think that is? Google's sheets are not quite Excel, are they? Isn't that strange? It ends at O. This ends at Z. I guess that's how Google does it. Anyway. Yeah, I'm not happy with my printer, eh? Alright, so basically I think we haven't quite separated the adults from the children yet, but the process there is an uphill battle. I think we have our hypothesis. Just to reiterate that it's unlikely that mindset will work because the 4 clinical trials GSK ran, 3 of them failed, and even if the very best one recapitulated, which is very unlikely, it still would have a good chance of not being statistically significant. And then finally, of course, idaloperidine did not work, so why should intepirdine? Those are my 2 theories, both of which spell doom, I think, for the company, or at least not the company, but the drug in that trial. What we did is we created a big literature index, which I think basically if you read all these papers and all these patents, there's almost no way that you wouldn't have a full understanding of this drug. And so we'll see if we can start to do that. What I like to do is I like to mark the papers down with these numbers. I'll just write 7424571 on top, and then when I'm done reading it, I'll write when I read it, the relevance and the topic, and then as I read them, I populate the model with the other sort of parts of the model, if that makes sense. So that's basically kind of my approach to this. You know, in addition to reading the company's press releases, we should also hear the company's conference calls, and then also talk to the company if we can. So anyway, that's about it. We'll be back with part 4 at some point soon. Thank you so much for joining.